Welcome back to The Explainer. Today, we're diving into a fascinating and incredibly high-stakes biological heist. We're tracking down a microscopic criminal that has basically mastered one of the most devastating, efficient robberies in human history, stealing the very water right out from our cells. And just as importantly, we're going to walk through the exact clinical lab investigation that modern science uses to actually catch it. Think of your body like a highly secure vault. You've got stomach acids, mucus barriers, and a heavily armed immune system standing guard. Yet somehow, this tiny, coordinated invader manages to slip right past all of our biological security, hijacking the vault from the inside out. So how does a single microscopic invader drain the human body of life-saving fluids in mere hours? To figure that out, we really need to examine both the crime scene and the forensics. Okay, let's dive into this. Here's our roadmap for today. First, we'll meet the microscopic invader. Then, we'll look at the anatomy of infection, followed by the immune response and the resulting chaos. After that, we'll get into detecting the cholera culprit, looking at advanced molecular diagnosis. And finally, we'll wrap up with a summary and some core clinical takeaways. Part 1, Meet the Microscopic Invader. Our prime suspect in this heist is Vibrio cholerae. This is a gram-negative bacterium, and it has this highly distinctive comma shape, giving it a slightly curved, rod-like look when you get it under high magnification. Now, this is the sole pathogen responsible for the disease known as cholera. Epidemiologically speaking, its main route of transmission is fecal-oral, usually hitching a ride through contaminated water supplies. It just thrives in aquatic environments, literally waiting for that perfect opportunity to be ingested by a human host and kick off its assault. Part 2. The anatomy of infection. Let's move along and see exactly how this builds into a full-scale invasion. It all kicks off at the portal of entry, ingestion. The bacteria get swallowed down, usually in that contaminated water we just talked about. Now, to survive, they first have to run the gauntlet of human stomach acid. The ones that actually make it through reach the small intestine, and that leads us right to step 2, colonization. They start multiplying rapidly. But, just floating around isn't going to cut it. They need a solid staging ground. So step 3 is adhesion. Using these specialized appendages, they firmly anchor themselves to the mucosal wall of your gut. Once they're locked in securely, they execute step 4, the final phase of the heist, unleashing their specialized toxins. And that brings us to the bacteria's ultimate weapon, Cholera Toxin, or CT for short. This is a remarkably powerful enterotoxin. But here's the crazy part. Instead of just directly killing the cells of the gut lining, the toxin acts way more like a master hacker. It binds to your intestinal cells and actually alters their internal signaling pathways. Specifically, it forces those cells to just constantly pump out chloride ions. And well, where electrolytes go, water is going to follow right behind by osmosis. So your intestinal cells essentially start dumping massive amounts of water and electrolytes directly into the gut lumen. The result of this biochemical hacking is honestly staggering. An infected person can lose up to 20 liters of fluid in a single day. I mean, think about that volume for a second. 20 liters. That's like emptying out 10 large soda bottles straight from your bloodstream. That kind of massive, rapid fluid loss leads to severe, life-threatening dehydration and massive electrolyte imbalances in just a matter of hours. Clinically, this manifests as the characteristic rice water stool that's a hallmark of severe cholera cases. The stakes of this biological heist literally could not be higher. Part 3. Immune response and chaos. The dark genius of the Vibrio cholerae attack really lies in a striking paradox, and comparing healthy and infected tissue brilliantly illustrates it. In healthy intestinal tissue, it absorbs water normally, keeping a nice, intact mucosal barrier. But in the infected tissue, you've got this massive fluid hypersecretion and severe dehydration. Yet, if you look at the cells physically, there is remarkably little structural damage. Unlike other bacteria that cause bloody diarrhea by basically destroying the gut wall, Vibrio cholerae just flips a biochemical switch. It totally evades a massive inflammatory immune response or tissue necrosis. It's quietly draining the vault without ever blowing off the safe doors. Part 4. Detecting the cholera culprit. Now we switch to the investigation. The clinical lab workflow operates on a really strict, escalating timeline of evidence gathering. It's basically a race against the clock. Step 1 is specimen collection, handling the stool sample with incredibly strict infection control precautions for obvious reasons. Step 2 takes us to the microscope to look for immediate, direct visual clues. Step 3 is the culture phase, where scientists actually try to grow the evidence on specific media over the next day or two. And finally, Step 4 brings an advanced molecular test to lock down DNA confirmation of the specific strain. Let's break these down a bit. The very first real-time lead a lab tech might get is through dark field or phase contrast microscopy, because Vibrio cholerae has a single polar flagellum, which acts like a tiny, high-powered molecular motor. It moves incredibly fast. Under the microscope, this looks exactly like a shooting star, with a highly characteristic darting motility. When scientists see these little microscopic shooting stars just zipping across their slide, it's a massive red flag that Vibrio cholerae is likely right there in the sample. But of course, just seeing it isn't enough. They need to grow it to be absolutely certain. The key to culturing this evidence is a highly specific environment called thiosulfate citrate biosalts sucrose agar. Yeah, that's a mouthful. So we're happily just going to stick to the acronym TCBS. So why this specific agar? Well, Vibrio cholerae loves alkaline conditions, and TCBS has a high pH that suppresses all your normal, everyday gut bacteria, giving the Vibrio room to thrive. On top of that, Vibrio cholerae ferments the sucrose in the agar, which drops the pH locally and turns the colonies this bright, unmistakable yellow. It is just an elegant piece of biochemical detective work. Part 5. Advanced Molecular Diagnosis. Now, what's really interesting here is the fascinating trade-off in the diagnostic toolkit. We just talked about culture, which is the gold standard because it isolates the actual living organism for a full profile. But the major limitation? Time. It can take a day or two. On the flip side, rapid diagnostic tests, or RDTs, are absolutely fantastic for quick screening out in remote field locations where outbreaks usually happen, but they do sacrifice some sensitivity. And then, sitting right perfectly in the middle, combining high speed with high accuracy, is PCR, or polymerase chain reaction. PCR is incredibly fast and sensitive, though, it does require a modern specialized lab setup. When labs deploy PCR, they aren't just looking for any random trace of the bacteria. They're looking directly for the genetic blueprints of its weapons. Multiplex PCR can simultaneously target specific genes, like CTXA, which actually codes for the cholera toxin, and TCPA, which codes for those adhesion appendages we talked about earlier. By detecting these exact DNA sequences, the lab definitively confirms not just the presence of the bug, but that this specific bug is fully armed and dangerous. It's basically the ultimate forensic proof. Part 6. Summary and clinical takeaways. Okay, let's close the case file on this one. So, the crucial point is that you've got to remember these core takeaways. The culprit is Vibrio cholerae, that gram-negative, comma-shaped bacterium. Its primary pathogenic mechanism is the cholera toxin, which totally hijacks cellular machinery to force severe fluid hypersecretion, all without destroying the cells themselves. For lab diagnosis, the gold standard is growing those yellow colonies on alkaline TCBS agar. And finally, the key molecular confirmation relies on using PCR to detect that CTXA toxin gene. We've seen just how effectively this pathogen operates and how diligently our laboratories work to identify it. But remember, this is an ongoing arms race. Pathogens are constantly mutating, constantly swapping genetic material. So the big question is, as bacterial genomes rapidly evolve, will our next generation of molecular diagnostics be fast enough and accessible enough to catch the next great biological heist before it spirals into a pandemic? It's a vital question for the future of clinical biotechnology. Thank you so much for joining me for this explainer. I hope it brought this microscopic investigation to life for you. Keep exploring, and I'll see you next time.